Hi everyone. Hi all. My name is Jess Bamford. Thank you all for having me here today and listening to me speak. And sorry I can't be there in person to come and actually meet you all. So I'm one of the founders of a project called Wild WebCap. So we are a fungal diversity survey project that runs in the southern Mount Lofty Ranges. And what makes our project a bit unique is that we don't broadly survey all fungi, but we focus on a specific genus called Cortinarius or webcaps, hence wild webcap. The diversity of this genus is not particularly well understood, particularly in Australia. So we're aiming to contribute to kind of unravelling what's present here and show why these native fungi are so important. So before I dive into telling about what we actually do at Wild WebCap, I'm going to tell you a bit more about this really incredible genus, and hopefully this will help you understand why I'm a little bit obsessed with it. So Cortinarius is thought to be the largest agaric genus of fungi in the world. So agarics are like your classic mushroom-shaped fungi, so they have a cap and a stem and they look kind of vaguely like what you would buy at a shop, like button mushrooms. It's estimated that there are between 2,000 to 3,000 species of Cortinarius across the world, and they occur in Australia, obviously, in New Zealand, the Americas, and across Europe as well. They are a mycorrhizal genus, which is sometimes referred to as a plant partner or a symbiotic fungus. So that means that they engage in a mutually beneficial relationship with plants where they share resources like nutrients and water. Fungal mycelium has this incredible ability to grow to really extensive lengths, provided that the substrate and the environment is appropriate. So mycorrhizal fungi are essentially able to expand the area that plants can source resources from in the soil. There are a couple of different types of mycorrhizal fungi. So Cortinarius are an ectomycorrhizal fungus, which means that when the mycelium meets up with the roots of a plant in the soil, it forms like a sheath around the root, and that's how their connection's made. Whereas there are also endomycorrhizal fungi which do a similar thing, but the fungus will actually penetrate into the root of the plant, so they have a different type of connection. And these relationships are formed with dominant tree species, with Cortinarius in particular. So in Australia, they have these relationships with eucalyptus trees, and in other parts of the world, they also form relationships with their dominant tree species. So you can imagine the immense diversity of eucalyptus trees that we have across Australia, and Cortinarius are kind of like their partner in crime. So they're like the eucalyptus of the fungi world, basically. They have that level of diversity and spread. But because fungi have this ephemeral nature and they kind of pop up for quite a short amount of time, in a lot of instances they're not particularly well understood, because you really have to be in the right place at the right time in order to see them. And seeing them is one thing, but seeing them under conditions where you can actually identify them is another thing as well, because sometimes you get to it and they're a bit, smushed and rained on and that sort of thing, and that's not particularly helpful. I should also add, on a purely aesthetic note, they look very, very cool. They occur in a lot of different colours. So in South Australia, you can find purple, blue, red, yellow, green ones. There are an awful lot of brown ones as well, but to be completely honest, the really huge number of brown webcaps isn't what really grabbed me and made me fall in love with them, but they are still really important nonetheless. So at Wild WebCap, we survey 6 different sites across the southern Mount Lofty Ranges in a variety of climatic conditions and ecosystem types. So from Parrawhilla up north to Deep Creek down south. And the long and short of it is that we go out and see how many we can find during autumn and winter. We record our observations and we take collections. I should add, you do need a permit to do that, which are being submitted to the South Australian Herbarium alongside morphological descriptions and information on the environment that they were found in, because we're trying to provide some more comprehensive data to go along with the specimens and the context that they actually occur in. Because this is such a huge genus, identification can be pretty tricky using just their morphological features. So we also use DNA analysis to help us confirm the identification of species and again provide supporting information for the collections. And this has helped us to confirm the occurrences of a few different species in South Australia that we weren't necessarily 100% sure if they were here or not before. So we've had that with a few different really cool looking red Cortinarius. They're called the Demosabe group or skinheads. And again, a lot of them look really similar to one another. So we had to do that DNA work to tease them apart and show that's actually what we had. We were really lucky to confirm the ID of a species which generally only occurs in southern Western Australia, and we didn't have any previous records of it in South Australia before, so that was super exciting. We also run workshops teaching people how to identify common species and how to upload records to iNaturalist to really harness the power of citizen science to help us better understand their distribution and their occurrence across South Australia. Some of the challenges that we have faced with this really come back to the lack of records and lack of genetic information on what we actually have in South Australia, which is what we're trying to help solve, really. We have many specimens that we couldn't find matches for on GenBank, or if they did have a match, it would be for unidentified species. So someone in Australia has found the same thing, but they don't know what it is either. So we're going, here's our thing, we don't know what it is, and it's the same as your thing that you don't know what it is, and that's not particularly helpful a lot of the time. But this is kind of the exciting thing about studying such a large group. I can't even begin to imagine how many species are out there and how many haven't been formally described. We also found that common species don't have many records that we can access across Australia, let alone South Australia. And the environmental records of where they occur aren't particularly robust either. So we will say, oh, they're with some kind of eucalypt, but we don't know what. So that's something else that we're trying to continue with as well. Another challenging part, I suppose, is that in particular this year, you all would have noticed it's been pretty dry, which doesn't make for good conditions for especially some of our larger fungi. Even though we've had some challenges, we still see a lot of value in this project, and we hope it encourages other people who are interested in fungi to kind of like adopt a genus or a species, and you know, you can really make a significant impact in your niche areas of interest if you just get stuck into it. So this is where you can find me. I'm most active on Instagram. I post ID guides, let people know about upcoming forays or workshops that we have. If you're not on social media, you can always email me and I can let you know when something's coming up. We're also in the process of making a local field guide, which is going to be out hopefully in 2026. So that's going to be like a short booklet showing you how to ID beginner-friendly species. But yeah, come along to the workshop and learn how to do it in person. They're always fun. I should also mention we have a really amazing fungal studies group in Adelaide. So there are a lot of great knowledgeable members in the group. We have our last meeting of the year next Tuesday. So if you want to come along, you can email me. We have a great new convener, which is me. So feel free to get in touch if you want to come along, and I'll send you the details for it. But that's me. Thank you.